Hello, everyone, and welcome to today's webinar, Using Single-Cell Multiomics to Accelerate Cancer Immune Cell Therapy Research. I'm Susie Valdez of LabRoots, and I will be your moderator for today's presentation. Now, I'd like to invite everyone to participate by submitting your questions during this presentation. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing this presentation, just click on the question mark icon on the left toolbar for further help or submit your concerns in the Ask a Question box. So let's get started. Today's webinar is presented by Dr. Adriana Suarez, Science and Technology Advisor at 10x Genomics. In this role, Dr. Suarez helps researchers adopt single-cell and spatial technologies to address their scientific questions. For a complete biography of Dr. Adriana Suarez, please click on the Biography tab at the top of your screen. Dr. Suarez, welcome. You may now begin your presentation.
Well, thank you for the introduction, Susie, and thank you everyone for joining us today. We are going to be talking about how single-cell multiomics is helping us tackle these very tough questions that we have in cell therapy research. So we have a lot of promise, but we also have fundamental questions that we need to address. So hopefully in this talk, you will get a view of how these single cell technologies are being used for cell therapy discovery and development. So first, I wanted to give you an overview of the field, basically the state of the art. And I wanted to start with this quote from Sam Behati, an oncologist from the Sanger Institute. He compares the technology of single cell with a microscope, how these platforms are giving us a whole new lens into life and biology, and it's giving us a new instruction manual so we can leverage that to really tackle these tough questions that we have in different biological and scientific fields. And of course, the topic of today is cell therapy research. In terms of what has happened in recent years, I wanted to give you an overview of some of the highlights. So in 2018, Science highlighted single cell as the breakthrough of the year. And this was more about the applications, how people and different research groups were using or are using these technologies to explore heterogeneity and in biology with a whole new lens. In 2019, Nature Methods selected single-cell multiomics as the method of the year. And so what we are talking about here is how we can start extracting different layers of information from the same cell. So we can go beyond gene expression and we can start looking at things like epigenetics, proteins, and other type of modalities simultaneously from the same cell. And last year, again, Nature Methods selected spatial gene expression as the method of the year. And this is a revolutionary type of analysis where we are actually extracting this information directly from tissues. So we don't lose that spatial context, which could be extremely important when we, for example, are asking questions about CAR T cells trafficking the tumor microenvironment. So we see that single cell is very complementary to spatial technologies. Of course, the focus today is going to be single cell, but I thought I will show you basically an overview of what has happened in the field in recent years. So at the next, we're really moving away from legacy tools like bulk. Bulk has been instrumental to help us address different issues in biology, but it has limitations. And especially in areas such as cell therapy, where we have a lot of heterogeneity and we have a product that is actually evolving over time. So with bulk, what we get is an average, and that average, it's basically biased towards the most common cell type in our sample. So we can appreciate the amount of information we are leaving on the table just because we don't have the resolution to gather this information in a more specific way. With single cell, we can extract information from each cell in that sample. So we get something like we see here, where each dot or circle represents a cell. And then we can start coloring these dots or cells based on their phenotypes. If we're looking at genes being expressed, at open regions of chromatin, at proteins, or a combination of all of these different modalities. So we can have a combination of on super biased clustering, basically let the data tell us what type of clusters we have, but we can add to that annotations with canonical markers to actually find the specific cell types or even cell states in our sample. And then with spatial, as we mentioned before, this is something that we are also developing at 10x. So we have, for single cell, we have our Chromium platform, and for spatial, we have our Vision platform, where we can look at these gene expression or even proteins from the tissues directly. And what we see is that research groups are actually using these platforms in a complementary fashion, so they can extract as much information as possible from their in their projects. And in terms of publications, at 10x, we really keep a close eye on what people are doing with this tool. And the best way to do that is just looking at the work that is being published. So we are over 2,200 publications right now. And of course, this is very exciting for us at 10x, but it's also a great resource for, I think, most people in this call to really see how these technologies are being used to tackle questions that are similar to yours. So we see that as an inspiration, really see how these could be used in innovative ways, but also it can be very practical. So we see resources here associated with sample preparation for single cell experiments, all the way to software tools to really get into the data and the story behind the data. So this is a great resource that we have for all these different areas of research. And the good thing is that single cell gene expression has been with us for a long time now. So we have, I feel, an example for almost every type of question at this point. In terms of the tools that we offer at 10x, so we really wanted to offer a streamlined workflow for our different solutions. So we have instruments, we also have reagents, and I wanted to mention here the software piece. So we offer software that are free of cost, and these include two different type of tools. One is pipelines, so we really can process that data that comes from the sequencer, so we don't have to basically start looking and putting together a pipeline. But these pipelines are ready for you to use, so you can process those raw files from the sequencer, and then you can end up with a visualization tool, which we offer as well. So we actually see here in the right a screenshot of one of those visualization tools that we can use to basically visualize, assess, and run different type of analysis from our samples. And at the next, we have an innovation engine that keeps evolving, and this evolves based on the ask from different researchers we work with. So we have different options, and what we do at 10x really is to connect with researchers to help them find the best tool for their scientific question. Today, we're going to focus on a number of those solutions. We are going to see the use of single cell gene expression. So this is one of our first products. We are on version 3, and we're going to see how we can use this to assess single cells, extract the whole transcriptome of these cells, but we can also have a targeted lens when we work with a specific gene panel associated with our scientific questions or experimental design. We also are going to talk about our single cell immune profiling, and this is extremely relevant in cell therapy, of course, when we are looking at immune response and we are trying to really re-engineer some of these immune cells. So this solution allows not only to recover gene expression, full transcriptome or targeted, but also allows to get the T cell receptors from those cells. So the key part is that with this information, we can start tracking clones. We can really follow them over time and see how they are expanding and what's the persistence of these specific clonotypes. And we are also going to see really briefly and talk really briefly about our new single-cell Multi-Ohm assay, where we can extract information from gene expression together with epigenetics, looking at open regions of chromatin. So we can think about this tool on how we can visualize these switches that are programming the genes that are being expressed and could help us explain all these different phenotypes that we see with the cells. So we are here to talk specifically about cell therapy research. And Emily's story is really a testament of how impactful these technologies could be to help patients. Emily was one of the first humans and the first child to have access to an experimental treatment at the time for her B-cell acute lymphoblastic leukemia. So Kymriah is now an approved CAR-T therapy, the first one approved by the FDA. And we really hope to see more of these examples in the future. But we also know that there are fundamental questions we have to address to make this a reality. And here I want to show you just a few examples that we can think of on the type of questions, fundamental questions we have to tackle to really have successful cell therapies. So I'm just going to mention a few of the ones we have here. In terms of tumor heterogeneity, we have a lot of questions around antigens, antigen development, and of course, of tissue toxicities that we see with some of these therapies. When we're thinking about expansion and persistence of these cells, we also can think about how we can have a lot of heterogeneity in the product. So we can actually exploit these to understand better those products and start predicting what's going to happen to patients after we make infusions of these products. And then we can also think about the insertions and the edits that we're making to these cells and the impact this has directly on the phenotypes and the expansion of these cells in the tumor microenvironment. So all these different questions that we still need to resolve, but now we have tools that could allow us to tackle some of those in a very unbiased and scalable fashion. So here I'm going to give you different flavors on how we've seen these technologies being used in cell therapy research. So we have the high throughput aspect, and we touched on this before on how it's not all about just looking at single cells and looking at their modalities, their molecules, but also doing this at scale, where we can do this for hundreds of thousands, even millions of cells in one experiment. So this could be extremely powerful to discover T-cell enhancing targets. And then, of course, we have the aspect of multiomic characterization. So as we mentioned before, we can go beyond gene expression. We can actually look at the modifications that we're doing to these cells. We can look at T-cell receptors. We can look at even proteins or multi-ohm or epigenetic characteristics of these cells. And in some cases, we can make these measurements simultaneously. And we're going to see examples on how this is being used in different points of development in cell therapies. We, of course, can exploit that information to identify cell states and cell types. And so we can use this information to uncover suppressors or activators that are cell type specific in the tumor microenvironment. Another aspect that we can explore is how we can start tracking this clonal behavior. And for this, it's very critical to have T-cell receptor information at a single cell scale. So we can actually see these clones and start linking phenotypes of these clones with their behavior. We can start using these tools to distinguish between CAR-positive or transgenic TCR cells from endogenous T cells. And another example here would be using unbiased gene expression detection to look at expression of antigens in unexpected places or cell populations. And again, we're going to see examples on how that's being used in the research. So I'm going to walk you through these examples with this type of structure, going from target discovery to characterization of the infusion product. Then we're going to see examples in response monitoring, and we're going to finish with one example in resistance profiling. So the idea here is that we give you a tour of all these different publications that we've seen in these different areas to advance cell therapies. So starting with target discovery, there are so many different directions that we can take here, but I want to focus on these two key questions related to target screening and functional screening. So when we think about functional screening, we could think about an experiment where we use a single cell gene expression solution from the next, and then pair that gene expression profiles from these cells together with DNA amplicons for specific modifications we've done to those cells. So with this information, we have paired libraries where we can actually look at the phenotypes and the modifications we are doing to those cells to see the direct link between those modifications and the effects they have on those cells. And so this is what this group from UCSF and Alex Marson's lab did. So they developed a new functional screening tool called pool knocking sequencing, or POCI-seq. And the idea here is that you start with a pool of knock-ins. So they started with 36 different constructs that you think will modulate T cell function. So for example, some of those will induce overexpression of genes associated with cell fitness or activation. And then you take these constructs and use them to modify a library of T cells. The next step is to apply selective pressures that mimic the environment that these cells are going to embrace in the tumor microenvironment when they are trafficking there. And then we use those cells with these modifications after these selective pressures to use the single cell gene expression profiling. And in this case, they extracted the library for gene expression, but they also used amplicon sequencing of these knock-in constructs to have that paired information. What is the effect of these specific modifications in the phenotypes of these cells? So one benefit of this approach is that we can start looking at the type of modifications these cells have. So we can have cells with one single construct, but we can also have cells with multiple modifications. So we can deconvolute that and really observe the type of modifications, if it is one or if it is a combination of more than one modification. And the most exciting part, of course, is looking at the phenotype of those cells. So what's happening with those cells in vitro or in vivo? And this is what this group was looking at. So they found that a specific type of constructs that were modifying or that were synthetic receptors at TGF BR2 lead to an increase in T cell abundance in solid tumors, but they also induce different cell states. One particular construct, 41BB, actually showed to promote accumulation of cells expressing key effector cytokine genes that will enhance tumor clearance. So very exciting finding and a very innovative approach to really look at this question in terms of discovering and doing targeted screens. The next example I want to show you, still on target discovery, has a totally different lens and it's more focused on target screening. So here we could think about using our single cell gene expression or even single cell immune profiling solutions to start looking at single cell atlases and see at the expression of specific antigens in places that we might not expect them to see being expressed. And so we can use this type of information to understand and really find the roots of tissue toxicities that we find with some of these therapies. So this is what this group from UPenn and Stanford, led by Carl June, Avery Posey, and Satpathy and others, did. They were focusing on CD19-directed CAR T cells. We know that these therapies have shown stunning efficacy in hematological B-cell malignancies, but we also know that we have major side effects associated with neurotoxicities, which could be lethal, like in most cases. So the hypothesis from this group was that perhaps there is a population of cells in the brain expressing the CD19 markers. And so some people were skeptical about this hypothesis, but they went on, they extracted data from single cell gene expression databases, focusing on prefrontal cortex cells. They excluded neuronal and erythroid clusters from their analysis. And to their surprise, they found a very rare cell type called pericytes expressing the CD19 marker. So they wanted to make sure this was not an artifact, so they did different subsequential testing. One of those was to look at the expression of other type of B-cell markers. So they look at the expression of CD79A. They found it in B cells, but they didn't see this marker in pericytes in the brain, confirming that in fact there is a population in the brain that is expressing the CD19 marker, being these pericytes cells. One question they had, of course, is a very comprehensive paper, but they also wanted to look at what happens in other organs, because we have pericytes in the lung, we have it in the kidney, but we don't see toxicities in these organs when we have CD19-directed CAR T cells therapy. So here we see the expression of CD19 in pericytes in the brain and in the lung. And in this green box, we see that the expression of CD19 is very specific for pericytes in the brain, but we don't see it in the lung. one potential explanation is the developmental pathways of these cells. So we know that parasites in the brain actually derive from different embryonic regions compared to those in the lung. So this could explain the different epigenetic pathways that they go through to end up with the expression of CD19 in the brain, but not in the lung. And so we can think about how this type of groundwork could be expanded beyond these CD19 directed therapies. And one comment from Avery Posey and other has been how with single cell, we really are able for the first time to identify this type of cell, this type of rare event, this small proportion of cells that, well, most people were not expecting to see in the brain. But it really comes down to the resolution that we have and the power that these technologies can allow us to have to assess heterogeneity. So as I mentioned before, we can think about how we could use this to de-risk cell therapy in terms of antigen discovery. So we saw this example on how we can use single cell to identify off-target toxicities, but we could also think about how we can use this to de-risk existing targets we have. And I think something that is very, very exciting is how we can think about the next generation of antigens in the pipeline or that we are discovering, and how we can use this baseline information to really build from there and start generating targets that are safer for patients. Moving on to the next aspect of development, I want to show you examples on related to characterization of the infusion product. So here we can ask questions about how this infusion product contributes to poor expansion or persistence and even toxicities of these therapies. And so here we can start combining, for example, information from the single cell immune profiling aspect together with clinical features to really see the impact of these products, their heterogeneity in the effects in patients and their response. So this work from Michael Green at MD Anderson did that. So they took the leftovers from infusion products that were infused to 24 patients. These are CD19 targeted CAR-T treatment. And then they used a single cell approach looking at global transcriptome, T-cell receptor sequencing, and they also used a targeted approach. So for this, they developed a panel of about 200 genes associated with immune response, toxicity signatures, and of course also CAR expression. And then they compared that data with clinical parameters. So their goal here was to assess, is there a connection between the characteristics of this product and the responses we see in patients? And they found that there were connections. So we see in blue the data from patients that responded to treatment, and in red, those patients that had poor response or progressive disease. So we see in blue these patients that progress. We see enrichment for memory CD4 and CD8 T cells. And patients with progressive disease, we see enrichment for exhausted phenotypes. So just to remember here that this is the infusion product. So this is characterizing these products before they could go into patients, could give us a good overview and potentially predict what the response is going to be. And this is another way to look at this data. So you will see that with single cell, you are gathering a lot of information. And so there are different ways to really extract signals from these data sets. So here we see different clusters that were found in the infusion product and how this relates to different patient response. So we see that cluster 1 was highly expressed in patients that had progressive disease. And here, the genes that we see, the signatures we see in the genes expressed is related to exhaustion and dysfunction. Cluster 4 is highly expressed in patients that responded to treatment, and then the signatures we see here in the transcriptome are related to central memory. Another interesting and really surprising finding here was the presence of a monocyte-like transcriptional signature in this infusion product. And the key part here is that they found an association between the presence of these monocyte-like cells with neurotoxicity syndrome. So the frequency of these cells with monocyte-like signatures were very, very rare. But again, we see how powerful these technologies could be to find these red events, tricky to find, tricky to expect, but that of course will have, and we see association with patient response. Moving on to our third area in terms of response monitoring, we can start looking at these tools and see how we can use them to see the evolution of the infusion product in vivo. And so here we have an example where we actually saw the use of single-cell immune profiling. So we can look at the gene expression, this phenotype of cells, together with VDJ sequences, and then start looking at these clonotypes over time. So this work from the Fred Hutch, look at that, really looking at the clonal dynamics of CAR T cells from patients over time. And so they started with a bulk assessment of these CAR T clones, and they noticed different patterns in terms of transcriptional profiles and evolution of these clones, but they couldn't come up with a specific association in terms of which type of clones are the ones being expanded and changing over time. So this is why they went to a single cell approach using the single cell immune profiling, and they were able to reveal different transcriptional profiles at different times during the IP product, at characterization point of the IP product, early, late, and very late after infusion. So we see that represented here in these different colors. And of course, we can start looking at the specific signals that are expressed at these different time points. So we see that early on, there is an enrichment of activation and effector response in these cells. But as I mentioned, the key part here is how we can use the clonotype information to see the type of clones that are actually driving these responses. So they used, using the single cell data, they focus on the infusion product and then identify 4 different clusters that we see here in these different colors. So cluster 2 showed a high expression of cytotoxic genes, and cluster 4 showed enrichment of proliferation associated genes. Cluster 3, on the other hand, show enrichment for genes associated with pre-apoptosis. So by looking at these clonotype information based on paired TCR sequencing, we can look at those clones, and then we can look at the gene expression and then identify what are the clones that are being expanded, driving these signatures. So we see that there is a progressive increase over time of clusters of clones that belong to cluster 2 and 4 that we saw had signatures of cytotoxic and proliferation associated genes. So it really shows how we can use this information to not only track these clones over time, but really see their phenotypes and their responses. A similar type of idea was used for the first in-human CRISPR trial, where we saw 4 different edits from these T cells that were being infused back to patients. So these different edits included changes to the TCR alpha and beta and PD genes, and also the introduction of an engineered TCR recognizing tumor antigen, the tumor antigen NY-ESO-1. So this group used the single cell gene expression platform and then captured the gene expression, but also the modifications that the cell had. So we saw this during a functional screening phase, but of course we see it now again in vivo. So as we saw before, with this type of analysis, we can start looking at the specific modifications these cells are going to have. So some of these cells are going to have 1, 2, 3, or 4 of those modifications in different type of combinations. So we can actually look at that specific data and all these different combinations. And so they really wanted to know what happens to these different mutations over time in patients. So then we see, for example, here at the time of the infusion, 10 days and 113 days after infusion, the differences between the persistence of these different mutations. So for the TCRA mutation and NY-ESO, we see that there is persistence over time. But for PD-1 mutations, these tend to become depleted. And then we can also look at the phenotype of these cells. So in this case, we're focusing on the NY-ESO type of mutations. And what we see is that these cells actually adopt a memory-like phenotype in these patients. So very promising aspect of this technology. And of course, this is going to give us a route and a roadmap for the next generation of cell therapies. And the last type of examples is associated with resistance profiling. So we can think about a different aspect that we haven't explored in terms of examples before, and it's looking at the epigenetic factors that are actually driving these different phenotypes that are wanted and those that are unwanted. In this case, exhaustion and dysfunctional T cells, of course, are phenotypes that we want to avoid. So what are the switches that actually are programming this type of unwanted phenotypes? And this could set the start to basically develop ideas to avoid them. So in this example, we're going to see the use of single cell gene expression to actually identify these transcription factors associated with exhaustion. But we could think about also using something like the single cell multi-ohm, where we actually are capturing not only the genes that are being expressed, but also the open regions of chromatin. So we can visualize this and start looking at specific repressors or enhancers of these gene expression programs. So going back to our example here, this comes from Rachel Lin's team at Stanford. And so they were really focusing on understanding the regulators of CAR-T exhaustion. So we know this is a barrier for the success of this type of therapies. So they created a model in which healthy T cells were driven to exhaustion by the expression of tonically signaling CAR. And then they, of course, explore all these different transcription factors that were driving these expression profiles associated with exhaustion. So one particular one called c-Jun showed to be very promising in terms of explaining this type of phenotypes. They found that when c-Jun is overexpressed, this renders CAR-T cell resistant to exhaustion. So it gives really interesting opportunities in the future to explore this particular transcription factor further. And I want to finish this example section with what's happening currently. How are these tools being used? And I'm gonna give you one example of that. So this is work from Nick Banovich at Tangent in collaboration with Christine Brown from the City of Hope. And they are using multiomic and multi-tissue profiling of CAR-T for glioblastoma. And so they are using a combination of different tissue and sample types they are collecting from patients, including PBMCs, tumor samples, CSF, together with the CAR-T product. And then they are applying a single-cell multi-omic approach. So they are planning to use the single-cell multi-ohm solution, the single-cell immune profiling together with the single-cell gene expression. And they are also adding this spatial aspect to their tissues to really look at where these genes are being expressed in the tissues directly and start asking questions about what happens when these cells are actually trafficking in the tumor microenvironment. And I want to finish with a last section on getting started with these single cell and spatial solutions. So this is going to be short. It's not comprehensive. It's just going to be an overview to get you thinking about these experiments. But just before I go there, if you have any questions, ideas on where to start with your single cell experiments, wherever you are, we have a 10X local team ready to talk with you and really help you address these experimental design questions that could be very important when you start thinking about single cell. So as I mentioned before, we have an innovation engine behind our solutions. So different options that can help you tackle your question in a very specific way. So the first step will be really looking at what's my goal and what's the best way to get there. And so we have a different type of options. And if you are thinking about multiomics, there are different ways to get multiomic information. But again, it's really based on your scientific question and what you want to get out of it. So the first step is selecting that solution for your question. And then the next step is, of course, thinking about all these different steps that you will go through when you're doing a single cell experiment. So everything starts with your sample, which is the input we see in the lab. So getting that sample ready, and again, this could be blood, it could be cell lines, it could be tumor tissue. So there's a lot of variation in terms of the samples that are run with our instruments. So really thinking about the best protocols to bring that sample to the experiment is going to set you to success. The great thing here is that we have so much data in terms of what works and what doesn't work for cell suspension generation. So we have a lot of resources that can help you have a really nice cell suspension for your experiment. The next step is library creation. So here we're actually using our single cell solutions and reagents that we selected in the previous step. And then you end up with libraries. So those libraries are going to have the single cell information of your molecules because they're going to be barcoded. They're going to have your cell address. And then we are going to be able to identify basically where they came from. So the sequencing steps can happen on Illumina platform. So our libraries are compatible with Illumina platform. And we have recommendations for all these different sequencing requirements for each solution that we have. And then the last step is data analysis and visualization. And I mentioned this before on how with Connect, you have access to pipelines, but also visualization tools that are open and free of cost. So with the pipelines, you can process that data from the sequencer and then get output files that you can use in our visualization tool, the Loop browser, or you can use third-party tools as well, which we have a lot, which is a great thing to have right now with single cell. And I just want to briefly show you what the workflow looks like for a spatial experiment with Visium. So here, of course, the input is different because we're working with tissues. So you will work with cryosectioning of your tissue. You will have a spatial set of slides that are basically, Visium is a set of spatial slides that allows to capture this molecular information directly from tissues. But we also capture the morphology. And this could be based on H&E or immunofluorescence, so you choose. And the same tissue section that you stain goes through library creation. And then those libraries are sequencing, and then we have data processing and visualization tools. So those later stages are actually similar to our single cell. But of course, the samples are totally different. So different perspectives on your samples. And so we have a number of resources. We have, as I mentioned, so many publications. We also have demonstrated protocols in-house, and there are customer-developed protocols that are extremely valuable for different types of samples. The last piece, and I think one of the most important ones, is the support team that we have. So always ready to answer your questions. We can be very specific, so please get in touch if you want to learn more. And I'm going to thank you, everyone, for attending. I'm happy to take any questions that you might have.
Thank you, Dr. Suarez, for that informative presentation. We will now start the Q&A portion of this webinar. And if you have any questions you want to ask, please do so now. Just click on the Ask a Question box. So let's get started. We already have some great questions coming in, Dr. Suarez. Our first question from an audience member is, what has been one of the most surprising findings in cell therapy research when applying a single cell lens to characterizing cellular products?
Wow, that's an interesting question. I think we saw a couple of those examples today. I think I'm going to pick two of the examples we saw, one related to target discovery and the other one in terms of characterization of the infusion product. So we saw in target screening, this group led by Ansasad Pathy and others, finding the expression of CD19 in a rare cell type in the brain, something that we were not really expecting. We knew we had neurotoxicities when we were thinking about CD19 directed CAR therapies, but using single cell, and it's really looking at the scale of these technologies, because finding these rare events, it's not easy if you don't have the capacity to really expand this to tens to hundreds of thousands of cells at a time. So really exploiting these cell atlases, they were able to find a specific cell population expressing this marker. And I think the most exciting part is how we can think about a similar approach to assess other type of antigens, and most importantly, to really develop the next generation of antigens that could be safer, because we could avoid some of these off-target and off-tissue toxicities. And then the second example we saw was more related to the characterization of the infusion product. So this work from Michael Green showing the presence of a monocyte-like population in the infusion product of a CAR product was very surprising. So we can start thinking on where this is going to take us when we start actually using these type of technologies in a broader context. And so I think the theme of these two examples and the other examples we've seen is heterogeneity, right? We're working with cell therapies, we're expecting heterogeneity, and now we have the tools to address it and to explore it. So we can use it in our advantage to address these really tough questions with the antigens we have, but of course think about how we can use them to redesign future ones.
Thank you so much. Now, are frozen tissue samples compatible with single cell RNA sequencing?
Yeah, so I mean, it's an important question in terms of how we can prepare samples for these type of analysis. Frozen tissue, it is compatible, and we have option in terms of how we can get into cell suspension or single nuclei suspension. So of course, the ideal situation is that we have fresh tissues, but we know that in a clinical setting, this might not be possible. So there are ways to preserve those tissues. If you have the option to bring those tissues to cell suspension and add a cryopreservant like DMSO, that would be ideal. But if your tissues are already frozen, there are protocols that could help us get to nuclei, which in those cases will be the ideal type of protocol to use. And we see that when we think about nuclei, this is very well-adopted workflow in those cases where we have frozen tissues. So yeah, we're going to have different ways on how these tissues are preserved. If you are able to talk about the logistics of acquiring these tissues, if you are in the designing stage of your experiments, I think bringing the next local team or people that are familiar with the technology will be extremely useful. So you design that collection and that processing of tissues. But if the tissues are there and that didn't happen before, definitely there are options for different type of preservation methods.
Thank you so much, Dr. Suarez. And I want to thank again our audience for these great questions coming in. Can I sort my cells prior to running through the 10x single cell assay?
Yes, absolutely. So sorting is compatible with 10x and widely used. So a lot of the examples we saw today actually sorted their cells, especially when we are looking at a specific cell population that we want to explore. That's the best option. If you're looking at single cell immune profiling, that should be your route. You're really sorting and reaching for a specific cell population and then going from there. That's not required though. So if you're looking at basically heterogeneity and then just looking at all these different cell types, perhaps in the tumor microenvironment, and you really want to assess what's happening there, sorting is not necessarily required. We also know that some people sort to really clean their cell suspension, so that's also an option. But there are also different ways to increase viability of your cell suspension. So yeah, it's compatible, but you have options in terms of how you process your samples before 10x.
Thank you so much. Now, our next audience member wants to know, why do I have to use nuclei with the Multi-Ohm assay?
Right. Yeah. So the Multi-Ohm assay, for those that are not familiar, we launched this product late last year, and we are very excited about it. I think a lot of the researchers we're working with are very excited and keen to start with this solution. It allows us to capture not only gene expression, but also open regions of chromatin directly from the same cell. And yes, the input is nuclei because we are after the chromatin inside the nucleus. So we found that the best way to do that, to access that content, is by starting with nuclei. One reason is that we want to avoid that mitochondrial DNA that we have in cells. So by removing the cell and just working with the nuclei, it gives us a better chance to do that. And we have optimized protocols to bring cells to nuclei suspension. And we also have protocols for different sample types. So we have 3 protocols, one for PBMCs and cell lines. We have another one for tissues that are easy to disassociate, like embryonic tissue, for example. And then another protocol for hard to disassociate tissue. So depending on your sample, we might have a protocol for you. And so there are options. And yeah, the reason for nuclei is just to get that chromatin accessibility information in a most efficient way.
Dr. Suarez, thank you so much. We have time for one more question. Where do you see single cell having the biggest impact in the field?
Wow, yeah, I think there's a lot of excitement about multiomics and really how these different modalities can help us understand these cells in a better way from a phenotypic perspective, but also from an evolutionary perspective, like how these clones are changing over time and how we can drive some of those changes by looking at these different modalities simultaneously. And I mean, this is in terms of technologies, the single cell multiomic aspect, and even including the spatial context, where you could think it could be another modality that you could add to your map of cells. And then in terms of areas of research, I think there is a lot of interest too on looking at the immune response of patients before they go to treatment. So we know that usually patients that go through cell therapy option have gone through different lines of therapies before that, chemotherapies that are altering these immune profiling in terms of their endogenous immune system. So I think there is a lot of interest in understanding that initial or post-treatment stage before they go into cell therapy, and then see how we can put all these variables together if we're working on an autologous type of approach, or even just looking at predicting on how these therapies are going to evolve and change within those patients. So I think that's an interesting research area. But as we saw today, there are so many different type of applications, so I'm sure we're going to see a lot of developments in the future.
Dr. Suarez, thank you again for your time today, for your presentation, and for your important research. And before we go, I want to thank our audience members for joining us as well and for their interesting questions. And a reminder, this webcast will now be available on demand through April of 2022. We hope you enjoyed this event. Thank you and have a great day. Bye-bye, everyone.